Hello. In this video lecture, we're going to go over chapter 20, which focuses on the lymphatic system and on immunity. Now, the lymphatic system consists of lymphatic vessels, nodes, and lymphoid tissue. We have to get some terms down. So first off, when I talk about immunity, that's the ability to resist infection and disease. The lymphatic system also maintains normal blood volume and the composition of interstitial fluid. When I talk about lymphocytes, these are primary cells of the lymphatic system. They respond to the presence of invading pathogens, abnormal body cells, and foreign proteins. Lymph is really interstitial fluid that's entered the lymphatic vessel. Lymphatics are the network of lymphatic vessels. They begin in peripheral tissues and end at the connections of the veins to the heart. So what you basically have is lymphatics that are sort of blunt-ended at the very smallest, farthest away part of the body, part of the heart, away from the heart. They're going to basically take up this fluid. Now, the lymphatic system includes an array of lymphoid tissues, organs. They're scattered throughout the body. Now, there's two major distinctions here. One is called primary lymphoid organs, and these are sites where lymphocytes are formed and mature. They include the red bone marrow and the thymus. Secondary lymphoid organs and tissues, these are sites where lymphocytes are activated. They are also cloned, and what I mean by cloned is identical copies of the cell are reproduced. These include lymph nodes, tonsils, mucosa-associated lymphoid tissue, which its acronym goes MALT, appendix, and spleen. All cells and tissue are involved in producing immunity. This is therefore called the immune system. This is a functional system, interacts with other body systems. Now, if you take a look here on this diagram, several of these, if you look on the right side, you will see the diagram showing lymph nodes. Those are those little clusters there. And you also have lymphatic vessels, which you can see spread out throughout the arms, legs, as well as going through the thorax, head and neck. Now, you're going to find that there are certain clusters that occur at the axillary and in the inguinal area. This is where you have a lot of the lymph nodes that cluster up and begin to do some of the refining, filtration, whatever that is necessary of lymph. You'll notice in the abdominal area that there are a variety of other lymphoid tissues, including the tonsil, the thymus, the spleen, the MALT, the mucosa-associated lymphoid tissues. Now, MALT is different areas, clusters that are distributed throughout the digestive, respiratory, urinary, and reproductive tracts. You will also find the appendix here. And of course, throughout wherever you have skeletal bone, you have red bone bone marrow. If you notice on the left side, you have a close-up image of what would be lymphocytes in lymph. Lymphocytes are different from other types of white blood cells, and they're distinguished by several characteristics. One of them is that they have a nice spherical or round nucleus. The other thing is that if you look at the total amount of space or volume within the cell, most of it is taken up by the nucleus. There's very little cytoplasm that is available, okay? And we'll get into the different types of lymphocytes, etc., in a little while. Now, interstitial fluid flows continuously into the lymphatic capillaries and exists, and thereby exists in tissues as lymph in the lymphatic vessels. Now, the lymphatic vessels carry lymph from the peripheral tissues to the venous system. Now, the lymphatic network will start at the lymphatic capillaries, which are the smallest vessels that you see in the lymphatic network. Lymph will resemble plasma, except that it's clear. Plasma, if you look at that, it would be somewhat yellowish because it has more suspended proteins, immunoglobulins, a variety of other proteins. Lymph itself has a much lower concentration of suspended proteins. Now, there is one exception to the rule. The lymph lacteals, these are the small capillary-like structures located in the villi of the small intestine. They are going to transport lipids that are absorbed by the digestive tract. So lymph there is going to have a milky appearance. Lymphatic capillaries are lined by endothelial cells, but the basement membrane is either incomplete or absent. Lymphatic capillaries, as I mentioned earlier, are closed at one end. They're larger than blood capillaries, but have thinner walls than blood capillaries, and have a flattened or irregular outline. Lymphatic capillaries have endothelial cells that overlap, which acts as a one-way valve. Virus, bacteria, and cell debris will pass into the capillaries, but it will not pass outwards. And this is partly due to these one-way flaps and the overflapping of the endothelial cells, as I'll show you in a few minutes. The lymphatic vessels typically occur in association with arteries and veins. So you'll see these vessels nearby what could be larger arteries, larger veins in various parts of the body. Lymph will flow from the capillaries to the larger lymph vessels. Since the larger lymph vessels also have one-way valves, they're going to, in essence, direct lymph toward the thoracic cavity. You're not going to have a backflow. And part of how this occurs really is the skeletal muscles, as they contract, they're going to, in essence, have an effect of squeezing on the lymph structures and cause a flow, similar to what you would see in the veins in the lower limbs, the legs. And basically what happens is you have this compression and it squeezes the lymph up to the next section of the lymph vessel, but it can't flow back because of the one-way valves. So if you take a look here, we have blood flowing through an arteriole and then getting into a meta-arteriole and spreading out into a capillary bed. But what leaves the capillaries is fluid, ions, and a variety of other factors, not huge molecules, not red blood cells. But what happens is that as the blood flows through this capillary bed, what would normally be brought back in is incomplete. And what I mean by that is that you have the recollection of a certain amount of fluid, but there's still a little bit of fluid that collects in the tissues. So what do? You see, if you don't process and get rid of that fluid, you're going to end up with edema, a collection of tissue fluid that occurs in the tissues and appears somewhat swollen, etc. This is where the lymphatic system plays a key role. Fluid will flow into the lymphatic capillaries, and because the lymphatic capillaries here are basically closed-ended, when you have certain compressions, etc., you will have the movement of this interstitial fluid, now called lymph fluid, and it will move in a certain direction. As you can see, as you have this movement of fluid, it is going to move further and further away from the point of origin, but closer, going into larger and larger lymphatic vessels. Notice that you can see a lymphocyte here, a lymphocyte here. Some of these cells will get drawn in. They will go through some of these one-way valves. And notice that some of the one-way valves are just basically where some of the cells kind of overlap one another, such as you see over here. So basically what happens is fluid would come in, but it cannot go back out. Furthermore, you have overlapping endothelial cells that create these one-way valves that are a little bit larger. And again, if you put compression in this area, fluid gets forced up, but it can't go back because of the one-way valve. It kind of collapses and closes off the direction. So all of the direction eventually is heading toward larger lymphatic vessels. And you can see this even in larger microscopic images where you see the formation of lymphatic valves as these lymphatics are passing along. This is an actual micro photograph image of a lymphatic valve. And so the fluid would go in this direction of the lymphatic vessel, all right? And you can see here in this slice of tissue, you basically have your artery, vein, and a lymphatic vessel really nearby. Small lymphatic vessels will converge to form lymphatic ducts, and these will empty into the subclavian veins. Lymph capillaries flow into either superficial lymphatics or deep lymphatics. Let's get into a little bit of the difference here. Superficial, you would think, oh, well, that's going to be closer to the layer of skin. Yes, they're located in subcutaneous layer deep to the skin. They are going to be found in areolar tissues of the mucous membranes of the digestive, respiratory, urinary, and reproductive tracts. They will also be found in serous membranes lining the pleural, pericardial, and peritoneal cavities. But what about deep? When we talk about this, they will accompany deep arteries and veins, thereby supplying nutrients and exchange of nutrients, gases, wastes, etc., to the skeletal muscles. Also, organs located in the neck, in the limbs, and in the trunk, as well as the walls of the visceral organs. So where do we get the drainage of this going back into the blood? Well, we have several ducts here. When I say duct, I mean D-U-C-T. The right lymphatic duct takes lymph and takes it from all of the right side of the body, superior to the diaphragm. So if you look at yourself and you have your left arm and you draw a line across where the diaphragm is and split it up from the midline upwards, you're going to get lymph that comes from right side of the face, neck, limb, etc. And that's where the right lymphatic duct takes the fluid and eventually puts it back into the blood. Now the left lymphatic duct, this is a little bit different. The lymph is coming from the entire body inferior to the diaphragm, so basically everything below the diaphragm, but it will also take the left side superior to the diaphragm. So again, the left side of the thorax, neck, head, arm. Let's show you that in a second in a diagram, but I want to go over a couple of other quick points. You have the right lymphatic duct, which is relatively short. It empties into the right subclavian vein. This is formed from the merging of the right jugular trunk, the right subclavian trunk, and the bronchomediastinal trunk. Now, the left lymphatic duct is much, much longer. It ascends along the left side of the vertebral column, collects lymph from the left bronchomediastinal trunk, the left subclavian trunk, and the left jugular trunk. So they all come together. Now, as we're moving up, we want to keep mindful of some structures below toward the bottom of the left lymphatic duct, and that is the cisternae chyli. It's a sac-like chamber at the base of the thoracic duct. It receives lymph from the inferior parts of the abdomen, pelvis, and lower limbs. Now, there is a term that you want to be familiar with also. It's called lymphedema. It's a condition where interstitial fluids accumulate due to a blockage of the lymph vessels. Most often affects limb where the tissue is swollen and stretched. Here we see the lymphatic vessels, the superficial ones, if you take and look here, at the lower area from the pelvis down to the beginning of the thighs. What you have are the superficial ones. They're below the skin, they're deep to the skin, but you can see where the lymph nodules are as well as lymphatic vessels. But if you go deeper into the tissue, you will see the deep inguinal lymph nodes located through here. Notice that they're right up against the veins and the arteries that will go and deliver blood into the respective basically thigh area going down to the lower limb. Okay. Now, as you move into the thoracic area, this is where we're going to get into right lymphatic duct, left thoracic duct, or left lymphatic duct. Right off the bat, as I said, from the right side, superior to the diaphragm, you're going to have this collection of lymph, and it is going to eventually come down from head, neck, etc. And it's going to come up from the right side where the upper limb is. It's all going to collect and eventually go in right here. This is entering the right subclavian vein, right at this particular point. Okay, these are the bronchocephalic veins. This is the right and the left internal jugular veins. Okay, and so what happens is just at that junction point, right about here, you have lymph from the right side of the body. Now, what about the left side? This is where you get all of the lymph coming in below the diaphragm into the lower limbs. On the left side of the thoracic side, you'll notice this thoracic duct that travels right on the left side nearby the vertebrae. At the bottom of it is the cisternae chyli. And so you have all of this fluid moving up, and eventually it will turn around and enter right in at this point here, the thoracic duct entering the left subclavian vein. Okay? Now, so you can see where the drainage occurs. You've got the right side, which really is almost like saying, well, it takes up only about 25%, just this part of the body, whereas the drainage from the thoracic duct, well, that's all here, both inferior to the diaphragm and then on superior to the diaphragm on the left side. Here's an example of lymphedema. So somewhere there is a blockage in some of the lymph ducts. So the tissue fluid, the lymph, will build up in the tissues here along the upper part of the foot, the lower part here, etc. And you can get some estimation there that some of the leg, the lower part of the leg, up around where the ankle, etc., is involved also. Now, what about lymphocytes? We've been talking about the fluid, but let's talk about the cells that are involved in the entire greater picture of immunity that occurs here. The lymphocytes are responsible for the immune functions of the lymphatic system. What antigens are substances that stimulate an immune response. Now, keep in mind, antigens are not just bacteria. They can be fungal cells. They can be parasites, helminths, roundworms, tapeworms, things like that. They can be protozoans. They can be also viruses, proteins in the virus coat. They can be also antigens from the perspective of select drugs, select food proteins, okay? Certain substances that will stimulate an immune response. So we have immunity types. We have cell-mediated and antibody-mediated. When I talk about a cell-mediated immunity, this is where lymphocytes directly attack the infected cells. And this is kind of important because if you look at the other type, which is antibody-mediated, otherwise referred to as humorally mediated, you have antibodies that are generated to attack, and what they will do is attach antigens, okay? So let's say you've got bacteria out there. Fine, they can have antibodies that are generated that will attach to the bacteria. But what if the bacteria hide inside the cell? Antibodies can't get inside the cell. So therefore, you have to have another attack approach. That's where you get into the cell-mediated attack. And with both of these attacks, whether the pathogen is inhabiting or basically existing inside of the cells, or it's transported and it's outside in the extracellular medium, you can definitely do an attack to neutralize it. Now, we have to understand clearly that there are different types of lymphocytes. These are the classes. We have T cells, B cells, etc. We'll get into those in a minute. But let's focus on T cells first. There are several different types. Cytotoxic T cells, these attack foreign cells or body cells infected by viruses. Do they include cancer cells? Yes. They will in certain cases attack cells that have gone cancerous or precancerous even. They will also attack cells that have been invaded by viruses, rickettsia, which are also simple bacterial organisms that hide inside of cells. Helper T cells will stimulate responses to both T cells and B cells. So they sort of play a role as kind of coordinator or manager. But helper Ts must activate B cells prior to B cell production of antibodies. So it's the one that sort of turns B cells on and says, okay, now start producing your antibodies. And there's a third group of T cells called regulatory. These modulate the immune response. With helper T cells, they control the sensitivity of the immune response. Regulatory T cells are believed to also play a role in controlling and thereby modulating the immune attack. So if there's no longer an antigen or a pathogen present, then regulatory will say, okay, guys, it's done. Cool it. It's believed that regulatory T cells play a role in keeping some of our immune reactions from going overboard and causing autoimmune reactions. That is, the immune system starts attacking your own tissues. Now, one other type that I have to include, but they actually show up in all of the other subtypes that I've just mentioned. These are called memory cells or memory T cells for the T cells. These respond to antigens previously exposed to, so they kind of hold back. They're on reserve if they have another exposure of the disease or the pathogen, etc. They get revved up again. They will clone, that is, copy more lymphocytes to repel the subsequent invader exposure. The other type we're going to talk about is B cells. They make antibodies. The antibodies will circulate in the body fluids. So that means blood, lymph. It will include also, some of the fluid that you see in areas like even the serous fluid, cerebral spinal fluid, etc. But basically, they are moving around in the fluids to defend against targets throughout the entire body. Okay? Now, plasma cells, these are the stimulated B cells that secrete the antibodies. Okay? Yes, B cells also can make a small grouping of their own cells, but in a memory form. So in other words, you can have memory T cells and memory B cells, and they wait for subsequent attacks later on. These are what is referred to, plasma cells are the ones that get stimulated, revved up, secrete antibodies. So everybody's clear on that. NK cells, which stand for natural killer cells. These are lymphocytes that attack foreign cells, body cells infected with viruses, and cancer cells. This is kind of borders between both the adaptive and the innate immune system. NK cells can be revved up by certain cellular signals. Now, the immune surveillance, this is continuous monitoring of peripheral tissues for foreign cells and or cancer. When I say foreign cells, I could be talking about bacteria, I could be talking about fungal cells, anything. Now, let's talk about the lymphocytopoietic lymphocyte production. That's what lymphocytopoiesis is, production of lymphocytes. First off, this will involve the red bone marrow. It will also involve the thymus, but I want to get into red bone marrow first. Red bone marrow will play a key role in maintaining lymphocyte populations because it produces immature T cells, B cells, and NK cells. Now, T cells, think of T cells as maturing in the T for thymus. The B cells mature to a degree in the bone marrow, okay? So immature T cells migrate to the thymus for maturation. Now this is via thymus hormone exposure and antigen selection. Now what the antigen selection is going on is T cells are basically being checked for the capability to target self tissue, which kind of sounds kind of weird, but let me explain it to you. Basically what is going on is that T cells will have a variety of T cells out there, and they'll have all these different targets. But what happens in the thymus is that they're sort of filtered. They're refined, they're processed so that none of the T cells can target your own self tissue, okay? Membranes, brain tissue, blood cells, nervous tissue, anything that belongs to you. These cells that have that capability are basically destroyed by what is called apoptosis. That's programmed cell destruction. This is a means to prevent, right off the bat, autoimmunity. In other words, your immune system attacks your own tissues. Now, in the thymus also, the lymphoid stem cells and descendants are isolated from the general circulation by the blood-thymus barrier. After maturation, the T cells will re-enter the blood circulation. Now, there are peripheral lymphoid tissues. We've mentioned a little bit before, but you have to understand that the B cells and NK cells will enter the bloodstream and migrate to peripheral tissues. Cell-mediated immunity basically comes down to T cells. Antibody-mediated immunity comes down to B cells. And immune surveillance, watching for any possible attacks, is done by the natural killer cells, NK cells. And so you see these classes here. Now, they don't mention as much in this text, but they will mention it if you take med microbiology, is that B cells will have a class of their own, memory cells. They will be on standby. And then if they find that antigen again showing up, let's say it's a protein from a bacteria or whatever, they will start revving up and they will start making copies of themselves, and some of the copies will get transferred over as plasma cells. As you see, what all these thin lines are is really the vast amount of endoplasmic and Golgi apparatus that shows up. So the endoplasmic reticulum, because what they have to do is make lots and lots and lots of antibodies, which are made of proteins, very quickly and in vast abundance. Okay? So, moving on. When we talk about the thymus, I've talked about this. This is where you do the filtering and processing for the T cells. They get prepared. And what you also have is that lymphoid stem cells will be oriented in such a way so that they will be ready to differentiate into their respective, whether they're cytotoxic T cells, helper T cells, or regulatory T cells. Okay? And in the red bone marrow, you have hematocytoblasts. Some of them are going to be the lymphoid stem cells. They'll migrate over to the thymus, and that's where you're going to have T cell maturation. The other types are lymphoid stem cells that differentiate into natural killer cells for immune surveillance and B cells for the antibody-mediated immunity. Hope that makes it a little bit clearer now. Now, lymphocytes aggregate, that is, cluster together within lymphoid tissues and lymphoid organs. Lymphoid tissues are defined as connective tissues dominated by lymphocytes. Lymphoid nodule, these are lymphocytes that are densely packed into an area of areolar tissue. If you remember what areolar tissue is from AP1, it's very loose. It's got lots of spaces in between. As a result, you're going to have no clear boundaries, no fibrous tissue, sort of like a capsule to enclose it all. Lymphoid nodules form large clusters. For example, the aggregated lymphoid nodules of the Peyer's patches. These are located deep to the epithelial lining of the intestine. You also have mucosa-associated lymphoid tissue, MALT. Okay? Now, these will protect the epithelia of the digestive, respiratory, urinary, and reproductive tracts from pathogens and toxins. Lymphoid nodules also have germinal centers for dividing lymphocytes. Lymphoid organs, though, differ because they're separated from the surrounding by fibrous connective tissue capsule. And it is the lymphoid organs that basically these include the lymph nodules, the spleen, and the thymus. Here you have an aggregated lymphoid nodule. In this case, this might be a Peyer's patch in the intestinal mucosa. So it's deep enough not to be in direct contact, but it does monitor if there is any presence of pathogens, etc. Okay, so it's in the intestinal wall, so that would be the Peyer's patch. But now what we're going to do is start going over some of the large lymph nodes in the pharynx. So we start talking about these larger structures here. And the first group that we're going to deal with is in the tonsils, in the nasal oral area. Okay? Basically, tonsils consist of a pharyngeal tonsil, there's one of those. A palatine tonsil, there are two of them, and a lingual tonsils. Tonsillitis is the inflammation of the tonsils, especially of the palatines, and these become infected and swollen. Now, tonsils will atrophy after puberty. The lymph nodes, these are the largest, are located in the neck, in the limb connection to the trunks. So what we're talking about is the areas of the groin, the axillae, and the base of the neck. Lymph nodes are slightly different in that they are kind of kidney bean shaped. They're often called lymph glands. The function of lymph nodes is to filter of lymph as the lymph passes from the afferent lymphatics. Remember, afferents are heading toward a particular structure. And what they do is you have the lymph pass through the subcapsular space, the outer cortex, the paracortex, the medullary sinus, and then they come outward via the efferent lymphatics. And if you want to know which one's which, real simple. For any lymph node, you'll have more afferent lymphatic vessels than you will have efferent lymphatic vessels. Here we have the lymphatic system as we talk about the pharyngeal tonsil. If you notice that there are a variety of germinal centers there within the nodes, and you have this pharyngeal epithelium. The pharyngeal is sort of in the back in this area of the pharynx, the naso-oropharynx. You have in the oropharynx area the palatine, and then the lingual, which is at the base at the base of the tongue. Now, here's a lymph node. If you notice the lymph node, it's connected by lymph vessels. Lymph may have repeated pathways where basically you're going to have repeated filtration occurring. So what you have is lymph coming in. Notice that you have the one-way valves here, and they deliver lymph via the afferent lymphatics, okay? These will then pass through the subscapular space, the outer cortex, the paracortex, and eventually the medullary sinuses, which is in this area here, and eventually out the efferent lymphatics. Notice that there the efferent is only like one, whereas the afferent you have many. Notice that you have the trabeculae, which is an extension of this fibrous capsule. So every one of these septa or trabeculae kind of have these germinal centers here. They kind of wall off almost completely, but not completely. But you end up with separation so that you can have these, basically these germinal areas, multiple ones throughout. And what happens as the lymph comes through this area, it will have the exposure to a variety of these cells, which will include, of course, B cells, T cells, things like that, NK cells. And so if there's any toxins, any antigens present that would stimulate immune reaction, this is how you're going to spread this over the area so that it would monitor for the presence of any pathogen or any threats, okay? Notice that there's a hilum right here, which is where you have the lymph node artery and vein coming in, as well as the efferent lymphatic coming out, okay? Now, the thymus is also a lymphoid organ, and it produces functional T cells. It is also an endocrine organ in a way because it produces thymosins. Now, these are the hormones produced by the thymus which promote the development and maturation of lymphocytes, basically T cells. After puberty, though, the thymus gland shrinks and becomes more or less somewhat fibrous, fatty. This is called involution. The capsule covering for the thymus exists, and actually the thymus is divided into really two lobes. The fibrous partitions are called septa. The lobes are divided into lobules. Now the lobes consist of the following: a dark outer cortex. This is where the lymphocytes divide and mature. Remember sometimes I talked earlier about cloning. So if they get stimulated, they have to start making lots of copies of themselves to go after a particular pathogen or a particular antigen. They have a light central medulla. Excuse me. Now the epithelial reticular cells These are cells that regulate T cell development and function. ERCs, epithelial reticular cells, also encircle capillaries to help maintain a blood thymus barrier. Now, if you see from the larger view here, this is where the thymus is in the thorax, superior to the heart, okay, in the mediastinum area, okay. Notice that you have sort of a right and a left lobe, and then the lobules. And if you move closer, you will see the septa, which separate out the lobules, right lobe, left lobe. Now, after about 3 weeks of processing, T cells will leave the cortex and enter the medulla of the thymus. T cells may exit the thymus via the medullary blood vessel or via the efferent lymphatic. ERCs will form these concentric layers called thymic corpuscles. They're also known as the term Hassel's corpuscles. And this is where function in the production of the T regulatory cells, okay? So it's a subclass of the T cells. Remember that? Here's an image. Okay, here's our thymus. We've taken a slice. We can see where the lobule is. There's another lobule here. There's a separation here, the septa, right here and here, okay? Here is the outer structure, the cortex. Here is the inner structure, the medulla. Now, increase your magnification, and what do you see? Lots and lots of lymphocytes all over the place here. You can tell by the darker stain. But some cells are staining very lighter, which is what you see from here and here. These are the epithelial reticular cells, okay? And this whole structure that has been made up by the ERCs is called a thymic corpuscle. Now let's move from the thymus to the spleen. It's really the largest lymphoid organ, responds to antigens in the bloodstream. What are its functions? To remove abnormal blood cells and other blood components via phagocytosis, storing iron that's recycled from the red blood cells, initiating immune responses by B cells and T cells in response to antigens in the circulating blood. Now, it's interesting to note that the spleen is attached to the lateral border of the stomach via the gastrosplentic ligament. That's important. You need to know that. We have also another hilum here. In this case, this is again an indentation. It's where the arteries and veins and lymphatic vessels enter the spleen. The visceral surface indents to conform with the kidney, the renal aspect, as well as the stomach, the gastric aspect. And I'll show you this indentation. This really is because it's a very soft organ. When we really look at this and you compare it to something, much more fibrous, much more solid, like a skeletal muscle, this is really a mushy organ, okay? And can rupture under certain conditions of trauma. And as it does, then you have to have a splenectomy, which is the removal of the remnants of the spleen. Now, the fibrous trabeculae, these partitions, they radiate outward toward the capsule through the interior from the ileum, okay? So the blood vessels travel within the trabeculae. The spleen is surrounded by a capsule containing collagen and elastic fibers. So here's the gross anatomical view. As you can see, there is a hilum right here where there are blood vessels. This, of course, is the stomach. This is the liver. This is the pancreas. Now, this is because the pancreas is partly retroperitoneal. There's the peritoneal space, okay? And you have the parietal and the visceral peritoneum of these various organs, stomach, liver, etc. And if you notice the spleen, which is right here, it's got an attachment, the gastrosplenic ligament here. And then the spleen also has an attachment here to the back wall, okay? Now, if you move from here to here, this is where the hilum would be. Superior, inferior, this is the renal indentation, and this is the indentation due to the gastric area. So the stomach would be up over here, and the kidney would be up over here. If you go back here, here is where our kidney is. Here is where the stomach is, And so the spleen has to conform to some of the shaping around these organs. As a result, you also have in the ileum the splenic artery, vein, and splenic lymphatic vessels coming in. Okay. Now, the spleen contains red pulp and white pulp. The red pulp contains a large number of red blood cells. It will also present macrophages scattered in the red pulp. Here's one point. If you look at a slide of the spleen and you get into the red pulp, you can easily tell the difference between red and white, because in the red pulp, there will be a number of erythrocytes, red blood cells present. This is the red pulp. It processes old and damaged red blood cells, pulls out the iron, destroys these red blood cells, okay? Now the white pulp resembles lymphoid nodules and contains lymphocytes. It will also present dendritic cells. These are antigen-presenting cells. You'll hear this a lot, APCs. It's basically the same thing as saying dendritic cells. They present antigens to teach the T cells and the B cells, hey, this is an antigen, this is a substance coming from a particular bacteria or a particular virus or whatever. This is what you have to look out for, okay? Now, the presence of these lymphocytes and dendritic cells and macrophages provide a rapid alert for infections in the blood. The blood flow through the spleen is a pathway of filtration for old blood cells and pathogens. If you take a look here, you're following the blood flow from the trabecular arteries, and these are basically branches of the splenic artery, which then eventually go into the finer branches, the central arteries, which are surrounded by areas of white pulp. The capillaries will discharge the blood into the reticular tissue of the red pulp, and this will also contain free and fixed macrophages. The blood will flow into sinusoids, whose walls contain fixed macrophages, and the blood collects in small veins that in turn merge to form trabecular veins that unite at the ileum. So what am I saying here? This is the direction of the blood flow through the spleen. If you look here, here's the trabecula. Here's a trabecular artery, a side view. Here is the central artery here. And now we get into this. Here's our red pulp. And I would say that if you increased your magnification, you'd see scatterings of erythrocytes, red blood cells. Here is your white pulp. Notice the distinction between red and white is how much stain to view the cells that the cells have taken up. Not much stain in here, a lot more in here. Now, innate immunity. I talked earlier and mentioned innate immunity versus adaptive, so we have to get kind of familiarized with the concepts a little. There are several defense mechanisms of our immune system that exist. The innate immunity is nonspecific, is not stimulated by specific antigens. So what am I saying here? It is not set up to distinguish between polio and smallpox, between streptococcus and staphylococcus, etc. It's not set up for the certain antigens, the proteins that might be on the outside of a poliovirus or on the outside of a protozoan or any of these other factors. It uses a defense mechanism that is either to prevent or slow the entry of infectious organisms or to attack them if they do enter. That's it. It's sort of the means to block and be the first layer of defense. Later on, adaptive is where we learn. So think of adaptive as learned immunity against certain pathogens, etc. Now, the defenses for the innate immune system are present at birth. We have a variety of them. Physical barriers such as our skin, the mucous membranes, the acid secretions of the stomach and urine. Phagocytes, these are cells that engulf pathogens and cell debris, so they would include things like macrophages and neutrophils, etc. Then we have immune surveillance. This is where our NK cells destroy abnormal cells. We also have some chemical defenses, interferons, These are defense chemicals to virus infections. Remember I said, hey, antibodies can't get inside of a cell. You've got to have some signal there. And interferon is one of these signals that says this cell is infected with a virus. Destroy it. Complement. These are proteins that assist antibodies to punch holes into the pathogen cells. Inflammation is a localized tissue response. And then we have fever. This is a body temperature elevation. You ever notice, okay, I've got the flu. I'm going to have a fever. Why? Because fever accelerates tissue metabolism and accelerates defenses. When you raise the body temperature, you also make it harder for certain pathogens to metabolically function. They may actually, at a higher temperature, start to have their proteins denature, as we've seen with what happens with denaturing. They may also slow down the effects of cells reproducing, such as bacteria cells or some other type of pathogen. Okay? Now, we have the immunity. First one we're going to deal with is innate. Later on, we'll deal with the adaptive. Physical barriers, phagocytes, immune surveillance, interferon, complement, inflammation, fever. Physical barriers prevent pathogens and toxins from entering the body tissues. The integumentary system, the skin, is a major barrier. Now, it includes secretions, which include antibodies, destructive enzymes like lysozyme, bactericidal proteins. One of them is defensins. We also have hair and stratified squamous epithelium. The mucus, which we see in the respiratory digestive system, the acidity of stomach acid and urine, and we also have tight junctions of epithelial cells and the fibrous basement membranes. So you can see all of this here. By the way, if this gets boring, think about it this way. How would your innate immunity function if you had third-degree burns over 30% of your body? If you look and review back to the first, the ANP1 course, one of the things that was mentioned in the integumentary system is the seriousness of skin burns where you lose that barrier. After the first 24 hours, the first 24 hours, the greatest risk of death, etc., is basically due to fluid loss. But after that, the big thing that everybody has to worry about is infection, because you've lost a major innate barrier. Okay? Now, let's go over phagocytes. They respond to pathogen invasion. Phagocytes include neutrophils, which circulate in the blood. They will enter the tissues that have injury or infection. They will phagocytize, in other words, engulf and bring inside cellular debris or invading bacteria. Eosinophils, interestingly enough, they will phagocytize foreign substances or pathogens coated with antibodies. It's an interesting test when you have a differential white blood cell count, and if they see a spike in the number of eosinophils, which their numbers are usually very small, in a normal person, when you see the spike, that's usually a warning that there has been an infection with parasitic worms present, because eosinophils usually take off when there's parasites present. And what I mean by take off is elevate their levels. Also, we have macrophages. These are derived from monocytes in the circulating blood. Monocyte macrophage system, otherwise known as the reticuloendothelial system, consists of fixed macrophages. They're permanent residents in specific organs and tissues. They are also scattered in connective tissue. So to help you, they will be present as Kupffer cells in the liver. They will be present in the spleen, etc. They don't migrate out. They stay fixed there, but they still have the phagocytotic properties. Free macrophages are mobile. They arrive at the injury site by migrating through the adjacent tissue or by recruitment from circulating blood. Here we have neutrophils. You can see that their nucleus is multi-lobed. Eosinophils is bilobed. The macrophages, you start off with monocytes and you either end up as a fixed macrophage or free-moving macrophage. Here is a phagocyte engulfing bacteria cells. Here's the bacteria cells here, and you can see the movement and the phagocytizing cell membrane wrapping around the bacteria. Now, just a couple of pointers, and this is a great chart to review. Phagocytes can leave capillaries by squeezing between adjacent endothelial cells. This process is known as emigration. All right. Phagocytes can be attracted to or repelled by chemicals in the surrounding fluid. This is a phenomenon known as chemotaxis. They are particularly sensitive to chemicals released by body cells or pathogens. And phagocytosis always begins with adhesion, the attachment of the phagocyte to its target. In this process, receptors on the plasma membrane of the phagocyte will bind to the surface of the target, kind of grab onto it, and then begin to wrap around it and pull it inside. After adhesion, the phagocyte may destroy the target itself, promote its destruction by activating specific defenses. Let's talk about NK cells because they are part of this immune surveillance and detection and destroying abnormal cells. So they're part of the innate system. Natural killer cells participate in the immune surveillance, which we've talked about. It's continuous patrolling peripheral tissues by NK cells. They can recognize cancer cells, and cancer cells are abnormal, usually by the presence of tumor-specific antigens. NK cells will destroy tumor cells but leave healthy cells remaining. The process has several steps: recognition and adhesion, realignment of the Golgi apparatus so that you produce a lot of secretory vesicles containing perforins. I'm not making this up. Perforins are very important because they can basically create holes in the cells that you want to destroy. What you're going to have is these vesicles filled up with these particular types of proteins and then transported to the cell surface. They will then be secreted, basically by an exocytotic method, and they will lead to the lysis of the abnormal cells. Now, there is a principle called immunological escape. This is the process of avoiding detection or neutralizing body defenses. This happens. This is where, if you go into medical microbiology, you deal with what's called host pathogen interactions, and some defenses by the pathogen can help evade some of these processes. Okay, so you can see all of this as it's described here. Step 1, recognition, adhesion. Step 2, realignment of the Golgi apparatus, basically firing up the system to make a lot of these perforin molecules, which will then form pores, and basically they will disrupt the balance of the fluids and ions by basically punching holes in the cell membrane of this unique or abnormal cell, and it leads to the lysis of the cell. Okay? Now, when we talk about NK cells, let's say we have a stem cell. It's gone through several divisions, continues through cell divisions, but there's a mutation in one of them. This mutation will follow itself down until it's detected by an NK cell. The NK cell will then act on it and destroy the abnormal cell. Now, if you sit there and say to yourself, well, golly gee, that's great, but how come we have so many people dying of tumors? Okay, what you have to understand is that when you have a cell that goes into the primary tumor, NK cells may not get all of those tumor cells, whether they're migrating yet or whether they're actually in the original tumor. Sometimes these progeny of these migrating tumor cells will later on survive. They'll pass through either the circulatory system, pass through the blood, and basically go into another area of the body, or they will go through the lymphatic system and go to some other area of the body and then begin to grow and divide. So this is where we talk about escaped cancer cells. Now, you might say to yourself, well, then why did the NK cells miss a few? It can happen where you don't have enough NK cells and where the tumor has been very difficult to get at, or the tumor may be highly aggressive. Usually when we say aggressive, it doesn't mean it takes people out and beats them up in a street corner. Yes, I'm adding a little humor here. But what it means is that the tumor is growing very, very quickly. Much more quickly than how the immune system, the NK cells, or any other cells can really mount an effective defense. And what happens is eventually, sadly, the patient will die. Okay? Now we have interferons in the complement system. They are distributed widely in body fluids. Interferons are small proteins released by activated lymphocytes and macrophages and by cells infected with viruses. So it's a means by which the cell may flag its neighboring cells, hey, I'm infected with a virus. Because you know what a virus will do. It'll infect the cell, take over the cell's machinery to replicate more copies of the virus, and then those viruses will leave that cell and infect other cells. Now, interferons bind to cell surface receptors and activate secondary messenger systems inside the cell. What is the result of this? You have antiviral proteins slow the spread of viruses, interfere with viral replication, and stimulate macrophages and natural killer cells. Really what interferons are a type of cytokines, which are chemicals released by local cells for cell-to-cell communication within the tissues. There are several different types of interferon. Yes, you need to know this. Interferon alpha, it is produced by cells infected with viruses. It attracts NK cells and enhances resistance to viral infection. Interferon beta is secreted by fibroblasts and slows the inflammation in damaged areas. Interferon gamma is secreted by T cells and NK cells and stimulates macrophage activity. So they all have a different approach to attacking invaders. Now the complement system, these are proteins found in plasma, okay? The function complements the action of the antibodies. I know that sounds goofy, but that's the way they brought up the name. Complement activation is via cascade of protein activation. To help you think about it this way, cascade. One acts on another, activates that, which acts on another, activates that protein, and another one acts on this protein. It is similar to what you're going to see when you start reviewing the clotting process with the various clotting proteins to form a clot that occurs in, let's say, an injury or a wound. But with complement, there are three possible pathways for complement activation. And there's a particular protein called C3b. Now, as I said, there was a bunch of different types of complement proteins. So you have several different ways to activate these proteins. And what C3b does is it enhances phagocytosis. So let's go through each one of them. There's a classic pathway, a lectin pathway, and an alternative pathway. The classic pathway, the most common, you have antibodies that bind to a cell wall to something. The lectin pathway, you have this unique sugar binding lectin called mannose binding lectin, and it binds to the bacterial surface because you will have mannoses on some of the bacteria surfaces, so this MBL will bind to it. So that's the flag to start the process instead of antibodies binding to the cell wall. Finally, they found another one called the alternative pathway, which complement proteins properidin trigger the cascade. Now, what is the result of all of this? First off, complement activation leads to the killing of the pathogen. There you have cell lysis. The membrane attack will damage the complex forms. You have cell holes that are forming in the membrane, and it triggers cell lysis. The cell just breaks up. You can also have enhanced phagocytosis. This is referred to as opsonization. The C3b enhances phagocytosis. It kind of flicks on and brings to the attention macrophages. They have receptors that detect and bind to complement proteins as well as to antibodies. Also, what happens is inflammation, and you end up as a result with histamine release, and this is from mast cells and basophils. These will accelerate blood flow and activate phagocytosis in the site where let's say there's an infection. So you can see all of these things. You can see there's C2, C3, C3b, C3a, etc. And they all act in this cascade format with eventually C3b binding to the bacteria surface and enhancing phagocytosis. That's the classic pathway. The lectin pathway is you have on the bacteria surface certain lectins, and you have this lectin that will bind to the bacteria cell wall because of the fact you have mannose there. Mannose is a type of sugar. So it's a monosaccharide. No, it's not a monosaccharide. I think it is a monosaccharide when I take it back. But it's a sugar that you need to know that, and this lectin will bind to it. And when that happens, it will trigger C3 activation. Okay, so you don't have C2, C4, and all these C1s and all this. Nope. It gets right to the heart of the matter, C3, which breaks off into C3a and C3b. And C3b will again bind to the surface and enhance phagocytosis. Now, the last one is a complement protein that interacts with plasma. And in the plasma is this properidin. It's also, you have several other factors, factor B, factor D. And what properidin does is it causes the C3 to be cleaved in half, just like in these two other cases. And what happens is that the C3a activates an inflammatory response. Of course, the C3b will bind to the bacterial surface again and activate phagocytosis. So what you've got are three different results: killing the pathogen, cell lysis, by forming these membrane attack complexes, basically these holes in the membrane. Enhancing phagocytosis, this is the opsonization. And finally, inflammation. So we talk about inflammation. What is it? It's a localized tissue response to injury. Now, fever is a generalized response to tissue damage and infection. Inflammation is a localized response to the injury. Now, inflammation produces cardinal signs and symptoms of inflammation. Now, there are four, but there's also a fifth one if you have lost a function. But the four that are key, and yes, you need to know this, are local redness, swelling, heat, pain, because it's a localized effect to the injury or the infection that occurs. If you have a fifth sign, the loss of function, let's say it's at my elbow, so I can't basically curl my arm or flex my arm, etc. It hurts, so I'll lose function of that for a bit. Also, I want you to take time to review innate immunity, okay? As you can see through this process right here. So we get into tissue injury, the chemical changes, mast cell activation, phagocyte attraction, redness, swelling, heat, and pain, and sometimes loss of function, and you end up with eventually tissue repair. Now, let's talk about fever for a few minutes. This is a body temperature greater than 98.6 Fahrenheit. If you're a Celsius person, 37 degrees Celsius. Circulating fever-inducing proteins, pyrogens as they're called, reset the hypothalamus body temperature set point. So this is all based on changing the hypothalamus. Now, for each 1 degree Celsius increase, you have a metabolic rate increase by 10%. Wow, that's a lot. And what you have is various pathways, which I won't spend too much time, but I want you to keep in mind a couple of pointers. The bacteria usually is going to produce either exogenous pyrogens, which are the bacterial toxins, and what will eventually happen is that everything focuses back down to the hypothalamic thermoregulatory center. Now, some of this may be carried along. If you'll notice, they have this basically inhibitory symbol right here for corticosteroids. Yeah, but you don't necessarily want to do that right now. What you want to do is get certain pyretics, the pyrogens, to activate the prostaglandins, which then prostaglandins will act on the thermoregulatory center. You increase the set point, heat is produced, you decrease heat dissipation, you have a body raising its body temperature, fever. Now, a couple points here. Enzymes that make prostaglandins can be blocked to keep this temperature up. What's the NSAIDs? Non-steroidal anti-inflammatory drugs. Okay, NSAIDs, aspirin, acetaminophen, okay, naproxen, etc. Okay, ibuprofen. All they do is basically block the enzymes that would make prostaglandins, thereby bringing down some of the fever. But there are some individuals that are called Darwinian, follow Darwinian medicine, and because they would ask the question, what was the survival advantage for having fever? And as we've talked about, when you increase fever, you begin to have negative effects on the growth of the pathogens, etc. You speed up metabolism, etc. So you get this sense that they don't want fever to go too high, but they don't want it necessarily be completely ablated, because then the patient may become overwhelmed by the infectious agent. Okay, this is the review of the innate system. Okay, what we're going to do is talk for a little bit about adaptive immunity. Not too long, but we need to go over this. Adaptive immunity provides the body's specific defenses. It's a coordinated response and is produced by T cells and B cells. Immunization, this is a means to induce immunity artificially. Just like right now, everybody is trying to find a vaccine for COVID-19. If you have that, you can induce immunity without the patient undergoing exposure. You see, you can get active immunity by either being exposed to the disease, the antigens in the environment, or artificially acquired by the administration of an antigen in the form of a vaccine to prevent the disease. The administration of the antigen teaches the immune system, when you see this, defend against it. Okay? And this response has to be both T cell and B cell oriented. You hear why some vaccines fail is because they provide a cellular response, but not a humoral response. What that translates to is the vaccine will rev up T cells, but it will not rev up B cells. Some will provide a humoral response, but not a cellular response. Usually when you want to have a good artificially induced immunity to a certain antigen, you want to have both present. Okay? Now, by the way, vaccines can be made from dead or killed, let's say, organisms, bacteria, viruses. You can kill them with using something like formaldehyde or formalin or heat. Or they can be a weakened pathogen that basically will not give the average individual a disease, but it will teach them, their immune system, to defend against it. There are other vaccines that are out there that are merely just proteins from the surface of a virus or the surface of a bacteria or the lipopolysaccharide of a bacteria. And these are also used to induce an active immunity. Now, I've been talking about active immunity for a minute. Let me talk about the passive immunity. So to help you think about it as a grid, okay? Basically, you can have active immunity, naturally or artificially, or passive immunity, naturally or artificially. Let's talk about passive immunity for a minute. This is naturally conferred by the transfer of maternal antibodies across the placenta, or by feeding the newborn breast milk that will contain mom's antibodies, and it will provide a limited period of immune defense for just a period of time, though, but it's enough to give the infant time to develop their own immune system and mature it. Now, artificially conferred passive immunity can be done by the administration of antibodies to combat infection. Maybe you might have heard of somebody getting gamma globulin or mixed IgG. Basically, what you're doing is you're getting the antibodies from somebody who had already been exposed to the disease, so some of those antibodies will be targeted for the particular disease. But again, antibodies will not last more than 30 days. Somewhere they may dribble along if you keep getting the exposure. But usually you get a shot like to prevent an infection of hepatitis A, because you may or may not have been exposed, they'll give you this gamma globulin shot. Now, it contains a lot of IgG molecules. They are oriented to attack Hep A, and they're great for the incubation period. They basically would attach to the hepatitis A virus, and it's a mean to kind of like sweeping out the immune system or sweeping out the body of the potential viruses, but it only lasts a short amount of time. It is not going to be something that'll be an active immunity. It's only passive, okay? Now, so this is what we have for immunity. We had innate, we dealt with, that's non-specific. Adaptive is specific. So if I give you a vaccine against hepatitis B, it is only for hepatitis B, not Hep C, not Hep E, or not Hep A, okay? You can get adaptive by the active immunity. It can be naturally acquired. You get the flu. Afterwards, you have antibodies against that particular flu virus. You can get it artificially if you get the flu vaccine. Your immune system will be prepped against the flu. Now, passive, produced by transferring antibodies from another source. Naturally, they get it in utero from mom's antibodies that cross over into the fetus's blood system. Or it can be if the newborn basically is breastfed. Artificially, well, there's a lot of people that sometimes they'll get like a spider bite from a black widow or a rattlesnake bite, and they're given these antibodies that will bind to the venom and will purge the venom, and they will be able to have some type of short-term immunity against the toxins from the venom of either the spider or the snake. Okay. Now, specificity. This is where we're going to get into a little bit of the issue of antibodies. The activation of specific lymphocytes and production of antibodies with targeted effects. Well, this depends on several terms that you have to be familiar with. First, versatility. Next, memory. Finally, tolerance. Versatility results from large diversity present in the body for this, basically this capability for the adaptive immunity. When exposed to specific antigen, the lymphocytes multiply to build a defense. All of the cells produced by the division of an activated lymphocyte, this is what you get into the formation of a clone. So in other words, you have one lymphocyte that is prepped to go against, let's say, polio. And it comes and is exposed to the polio antigen, basically a protein on the surface of a polio virus. You will then start forming multiple copies of the same lymphocyte to defend against the polio virus. Memory. You always have two groups of cells produced, one to attack the invader and one to remain inactive to provide reserve cells for later attack by the antigen. Remember, I said this earlier, you have memory cells that are produced for B cells and memory for T cells. Memory cells enable the immune system to remember the antigen it has previously encountered and will provide longer lasting counterattack. Tolerance. Lymphocytes have to ignore self-tissues. In other words, they cannot start acting on your own tissues. That's a bad thing. That's where you're going to get into autoimmunity. So basically, you have to be able to target abnormal and foreign cells and classify them as non-self. But self-tissues, you've got to deal with. Now, tolerance will develop over time in response to chronic exposure to an antigen in the environment. This is where you get the unique capability to start teaching the immune system, and this happens in a few cases when they try treating individuals for, let's say, cat fur allergies and stuff like that. They try to induce sort of a tolerance so the immune system doesn't react severely against some of these antigens, okay? Such as cat fur, ragweed, dog fur, horse hairs, or a number of other allergens. So, properties of adaptive immunity: specificity, versatility, memory, tolerance. Now, when we talk about adaptive immunity, and it's triggered by exposure to T cells and B cells to specific antigens, we have to keep in mind that specific antigens may stimulate A, the cell-mediated immunity via T cells, B, the antibody-mediated immunity via specific antibodies from activated B cells. We've been talking about that. Yeah, I'm going to keep reminding you because these are key points. Now, antigen presentation. How is it that the T cells and the B cells find out, hey, you see this protein here? Well, this came from basically Borrelia burgdorferi. Okay? What? Lyme disease pathogen. Oh, okay, fine. Now, sometimes vaccines are easy to make. Other times they're not. So you need to be aware of that too. But if you have a particular bacteria or a particular virus, how do you make the appearance of the antigens, the fragments of that show up in the plasma membranes? And there's a particular system that involves something you may have heard of before: major histocompatibility complexes, MHC proteins. You're going to have two classes, one that's present in nucleated cells of the plasma membranes. This is about all the cells. The only cells that you don't see it in is really red blood cells. Two, the class 2 is present in cell membranes of antigen-presenting cells. Remember that? Dendritic cells, etc. And lymphocytes. Now, antigen-presenting cells, these are APCs, specialized cells that include all phagocytic cells of the monocyte macrophage group and the dendritic cells of the skin and lymphoid organs. By the way, dendritic cells in the skin? Question mark? Yeah. If you go back to chapter that is on the integumentary system and you look at the very lower levels of the epidermis, you're going to have a cell called the Langerhans cells. Okay? Not the islets of Langerhans cells. Those are your dendritic cells in the epidermis, in the lower parts of the epidermis. Comes back here after a while. Yep. So we have adaptive immunity. Antigen presentation triggers an immune response. It has to be cell-mediated as well as antibody-mediated. The attack by antibody-mediated by B cells will be circulating antibodies. The direct attack, chemical or physical, will be from activated T cells. They will either find the pathogens and attack them by phagocytosis or release of chemical toxins. And the eventual end is the destruction of the antigens. Now, antigen-presenting cells, an antigen presentation occurs when an antigen glycoprotein combination appears in the plasma membrane of a cell, which will activate a T cell. Huh? Yeah. If you get a phagocytotic cell and it goes glump and begins chopping up the pathogen or whatever, it'll take fragments, and these fragments will be the antigens. They'll be attached to glycoproteins. The glycoproteins are genetically determined and are attached to the fragments of the antigens, as I said. And you will see this with MHC. It'll present all the time. But with MHC, that's MHC1, excuse me. But MHC2, the proteins appear only when the cell is processing the antigens. So to make this somewhat straightforward and simple, as if it could be, don't get overwhelmed, you'll get it. You have, for example, here, a pathogen, viral bacterial, triggers antigen presentation by class 1 MHC proteins. The infection results in the appearance of abnormal peptides in the cytoplasm. The endoplasmic reticulum produces class 1 MHC proteins, and the abnormal peptides are attached. Then we start exporting them. And we have this nice little export system via the Golgi apparatus that will take in vesicles the MHC proteins with the bound abnormal proteins, and there'll be sort of like a flag on the surface of the plasma membrane saying, attention, this has been found, this is bad. And if you think I'm making this up, you can see this image down here where a lymphocyte and an APC are having an exchange here. Here you have a phagocytotic cell, as I mentioned before. They engulf the extracellular pathogens. A lysosome will begin chopping it up because of its enzymes that are in the lysosome, and you end up with these fragments. You will have the endoplasmic reticulum produce class II MHC proteins. These fragments are going to be bound to the MH2 proteins, and they will be set on the surface of the plasma membrane. Now, infected cells stimulate the formation and division of cytotoxin T cells, memory T cells, and regulatory T cells. Okay, which is important. Inactive T cells are programmed to detect specific antigens bound to MHC1 or MHC2 receptors. If the T cell and the antigen match, the T cell and the antigen MHC complex will bind. This is called antigen recognition. Okay, so I talked with you in the tail end about all the bad things the T cell will do. It's kind of like saying, this boxer will beat the snots out of anybody else, and they will do this, this, and this. But then how do you get the boxer to the boxing match? The boxer has to know who's the bad guy to beat up. The boxer also has to know, how can you tell who's the bad guy? This is where we're teaching the T cells. This is the antigen, this is your target, and this is what you have to do. Now, I said this is antigen recognition, okay? Membrane proteins involved in antigen recognition are a broad class of proteins called CD markers. Now, what CD stands for is cluster of differentiation. It's like sitting there and saying, okay, fine, I got 52 cards, okay? And I start from the 2 all the way up to 10, and then I have Jack, King, Queen, Ace. But there's 4 sets. So you got to have some differentiation here. So CD markers are differentiations. They're usually also followed by a number. When HIV became really a focus of a lot of research in the 80s, suddenly everybody knew the buzz terms, CD8, CD4, etc. Basically, these key markers, CD8s, are found on cytotoxic T cells and regulatory T cells. They respond to antigens presented by MHC1 proteins. Okay? CD4 is found on helper T cells. They respond to antigens presented by MHC2 proteins. Okay? Here's my inactive T cell. It's not been awakened yet. Here's my infected body cell. It could be the APC. Here is an antigen recognition protein. Here is the T cell receptor. Here's the antigen. And here's the MHC here. So when these all connect together, this goes from inactive T cell to an active T cell. I mentioned the CD markers. So let's move in to antigen recognition. This prepares the T cells for activation. All right, but what about, do we have full activation yet? No, full activation requires exposure of the cell to specific physical and chemical stimuli. In other words, there's a couple of other signals that need to go on. But this is good because it also prevents errors, and these errors can turn out to be attacking your own tissue. In other words, autoimmunity. So let's talk about full activation. T cells proceed from recognition to activation by binding to the T cells binding of the T cells to a second site on the antigen presenting cell. This is called co-stimulation. Prevents the triggering of T cells to self tissues. Okay? Following the activation, the T cells start dividing and dividing and dividing and dividing. And they differentiate into the following classes. Memory T cells. Remember, memory is inactive. They go on reserve. They're out there for future use. Regulatory T cells. They control the responses to both B cells and T cells because they are going to secrete suppression factors. That's where I said before the regulatory guys are sort of like Okay, no more antigens here. Time to disperse. Everybody calm down. Regulatory T cells are also involved with time delay. A regulatory T cell will act after initial immune exposure. And you can see how all of this is set from antigen recognition to activation. The co-stimulation activates the CD8 T cell. And now we start dividing: cytotoxic T, memory T, regulatory T. Following the activation, the T cell will divide and differentiate into the following cells. I've talked about this. One big one, cytotoxic T. They're out to seek out the antigen-bearing cells. They will destroy the cells by perforin release. Cytokine release will cause apoptosis in infected cells. They will release lymphotoxin, which will also disrupt the cell metabolism. That's great. That's what you want, particularly if you're seeing something like a virally infected cell. And because of the virus, the cell is going to go cancerous. Okay? So you see how these are all set up here. And the three results. Perforin, basically, you're going to have lysis of the cell. Okay? Apoptosis. Cytokine release, that's, of course, going to cause apoptosis. And disruption of the cell metabolism through the release of lymphotoxin. Okay, now let's talk a little bit more about APCs. They can stimulate activation of the CD4 T cells, producing helper T cells that promote B cell activation and antibody production. Here's one of the clues. What I'm basically saying to you is that a certain subset of the T cells have to go over and kind of nudge the B cells. Hey, wake up. It's time to attack. Time to get all revved up and start making lots and lots of antibodies. But before CD4 T cells are activated, they must be exposed to antigens bound to the MHC2. Ooh, and again, we have to have also co-stimulation. Once the CD4 T cells undergo a series of divisions and differentiation, that results in an active helper T cells, which secrete cytokines that stimulate cell-mediated and antibody-mediated immunity. Memory helper T cells, which are inactive until you have subsequent exposure. And what I mean by that is these are the guys on reserve. They're waiting for later on in reinfections. You will also have sensitization of B cells. They will be prepared for activation by exposure to the antigen. B cells process antigens and present on the outside of the B cell attached to MHC2. So here's the antigen recognition of the CD4 T cell. All over there, co-stimulation. But this is the helper. So the helpers are going to be activated. And they're going to start releasing cytokines. And they're also going to make a group of memory T helper cells. Okay? You have sensitization. You have the antigens here. You have an inactive B cell. When the class 2 MHCs here, you have this binding to the antigen. The antigens are bound also to antibody molecules. One of the things you got to understand also is on the surface of B cells, even inactive ones, they will have some of their own antibodies kind of hanging out there because they're going to monitor for the presence of any antigens. Once you have the antigens bound to the antibody molecules, you will now get a sensitized B cell. Now, B cells, I said, are fully activated by binding to helper T cells activated by the same antigen. Okay. At the binding of the helper T and the B cell, cytokines from the helper T cells fully activate the B cell. So T cells have to release sort of these cytokines to get the B cell revved up. Once the B cells are revved up, they divide, they differentiate, they produce antibodies specific to that antigen. Now, these differentiated B cells become memory B cells, plasma cells. Plasma cells are the revved up, activated B cell. that basically differentiate to make gobs and gobs, incredible amounts of antibodies to that antigen. Memory B cells, there are reserves. They're going to be inactive. They're going to remain in reserve for subsequent antigen exposure. And upon subsequent exposure, these cells will convert to plasma cells and release antibodies. So here's part of the point. When you start talking about vaccination, okay? Now, because you got a vaccination against measles, let's say, measles, mumps, rubella, that's the one of the triplet vaccines. Yesterday, you could have been exposed to measles, and guess what? You're not sick. 2 years later, guess what? You're not sick because some of your memory Bs have been on alert. I shouldn't say on alert, but basically inactive, but remain in reserve for subsequent exposure to, in this case, the measles virus. Whenever it shows up, they start pumping out antibodies and they start basically binding to the virus and neutralizing it. Okay? That's to give you some perspective there. And we can see how this goes: B cell activation, division, differentiation with antibody production. Now, let's talk about antibodies for a few minutes. Not much, but it's important. Antibodies are small, soluble proteins. What I mean by that, they dissolve and they will be in a watery environment, etc. And they bind to specific antigens, and the abundance increases upon late antigen exposure. Keep that in mind when we talk about booster shots, okay? Antibodies are composed of two parallel pairs of polypeptide chains. You have one pair of heavy chains, okay, and one pair of light chains. Each chain contains variable and constant regions. And what I mean by variable is particularly they can change some of the sequences and some of the amino acid arrangements, so they will have the binding sites specific for the antigen. Constant regions of the heavy chains form the base of the antibody. The antibody binding sites, or the antigen binding sites, these are located on the free tips of the two variable segments, and the binding is specific for the antigen target. The antigen-antibody complex, the antibody will bind to the corresponding antigen molecule. And an antigenic determinant site, this is specific exposed site of the antigen that the antibody binds to. What am I saying? Look, up here is the antigen binding site. So part of it's composed of the light chain, part of it's composed of the heavy chain. Right around in this area, this is going to be parts that are going to be pretty much the same. They're not going to be variable, they're constant. These up here are variable. Why? Because what you want is having variation, and if you get into the genetics of this, it's mind-boggling. The guy that understood it all, that was able to write up and study it and understand it all, got the Nobel Prize, of course. Okay, it's great when you can sit there and say, well, these are the V genes, and it switches around the genes to make a particular unique antibody binding site for that particular antigen. Okay, so you'll get certain chains that have loops in them, and they will have disulfide bonds, but that's not really the key point here. The constant region here is constant, doesn't change genetically, because it contains binding sites that can activate the complement system. It contains binding sites that attach the secreted antibody to the surfaces of macrophages, basophils, and mast cells. Now, as I said, the variable region here, this is the one that's going to attach to a specific antigen. Notice the overall Y shape that you have here, okay? Now, let's say I don't have a big molecule. Let's say I've got a smaller one. Well, what's the difference? It plays a role in the antigenic determination site. With a complete antigen, the antigen with at least two antigenic determinant sites, one for each of the antigen binding sites of the antibody. Okay, that's a complete antigen, yay. But what if the molecule's smaller? This is where you get into what's called haptens, or partial antigens. Normally, they don't cause B cell activation. They must be attached to a carrier. This is where you have to match up a larger molecule with the hapten and attach them in some way, okay? Once you have this attached to the carrier, the antibody will attach to both the carrier and the hapten molecule. And this accounts for allergies to penicillin. This will also account for why we have what's called tetanus and diphtheria toxoid vaccines. You see, we're not looking for tetanus bacteria nor for diphtheria bacteria. We're looking for the toxoids that they make, which are somewhat incomplete. Sometimes what we actually do with certain other smaller haptens is we'll attach them to tetanus toxoid, and that way we'll get an antigenic response, and we'll get the immune system to go after both the hapten. It's already been set up to react to the tetanus toxoid, okay? Exposed surface of large pathogens has many antigen binding sites. And that's where you get into these images where you see like one particular large bacteria, and it's carpeted with antibodies. Here's the idea, okay? Complete antigen here. You got antigen determinant sites, bing, bing, and it attaches to the antibody. Yay, you got them. A hapten or a partial antigen. And it doesn't fit as well. It maybe only goes to one of these. That's where you have the larger carrier molecule here. You attach them together, you conjugate them as it's called, and they're able to create a complete antigen antibody complex, and then you will have a form of adaptive immunity. Okay, now here's what I'm talking about. Because you have, let's say, lots and lots of antigenic determinant sites on some bacteria or fungal cells or whatever, you're going to have different types of antigens here, and so you will end up with the possibility of having multiple antibodies specific to this antigen or this antigen or this one, etc. Okay? Now, let's talk about antibodies a little bit more. There are 5 major classes of antibodies, immunoglobulins. Now, the term immunoglobulin, they also put down IGs. I didn't put the small case S, but basically they all start off with IG, meaning immunoglobulin. There's immunoglobulin G, immunoglobulin E, D, M, A. IgGs, 80% of all antibodies are in the class IgG. They're responsible for resistance to many toxins, viruses, and bacteria. By the way, IgGs are the only antibody that passes across the maternal, fetal, placental barrier. So when mom is giving some of her antibodies to the infant in utero, inside the uterus, they're all going to be IgGs. Now, IgE attaches to exposed surfaces of bacillic and mast cells. IgD it basically is seen on the surface of B cells, and the binding plays a role in B cell activation, IgD that is. IgM, this is the very first antibody secreted after antigen is encountered. And what happens is you have this seesawing effect. After a little bit of time, IgEs start taking off in numbers, in the measured amount, and IgMs will decline. IgA is located primarily in glandular secretions such as tears, saliva, mucus, and semen. You can have several responses of antibodies. Here's how it goes. The primary response, this is when you first are exposed to the antigen, to basically the pathogen. You're going to have a time lag, and this is due to B cell activation. If the antigen is removed, the antibody levels will decline. But the secondary response, not the first one I just told you, the secondary response, the antigen is encountered again, and the antibody response is faster, greater in the antibody production. You will have also the fact that you're going to have memory cells respond. Memory cells will be converted over to plasma cells, and they're going to be cranking out lots of antibodies. Now, when we talk about memory response, this may be functional for up to 20 years. This is why you get situations where, for example, I was vaccinated many years ago for hepatitis B. And Hep B, you got a series of 3 shots, okay? You got day one, you get your shot. 30 days later, you get a second one. And usually by that time, about 96% of everybody's seroconverts means that they will now produce both cellular and humoral-based immunity. But to cover everybody, after 90 days, they get a third shot. And basically that's it. Now, you're supposed to get a booster. And 5 years later, I went to get a booster. And I got a booster. But 5 years after that, the doctor sat around and said, well, now what we do is we changed it. What we do now is we do what's called a titer test to see if you still have the the memory response. And if you do, you don't need a booster. And after that, I never needed a booster. So I'm good for the rest of my life, pretty much, for hepatitis B. Now, this is different for some other vaccines. For example, tetanus. Usually you have to get this every 10 years. So you got an injury, it's been 7 years, the doctor will say, well, we're just going to give it to you now to be sure. Usually a lot of other vaccines you're supposed to get at a certain point in your life. Everybody thinks it's a kid's thing, and they don't follow it up, which is why you start having situations of tetanus increasing in probability based on age. It continues to increase 30s, 40s, 50s, 60s because they never got another tetanus shot. Higher risk of having mumps and measles and a few other things later on in life if they've never followed up with a subsequent vaccination. By the way, in the primary response, you're going to have first the increase in IgMs and then the slow buildup of IgGs. In secondary response, you get a little bit of a bump of IgM, but you get this massive swelling amount of IgGs. So that's how it differs. If you'll notice, there's an importance to IgM. If you look at the tips of every one of these sections of the antibody, those are binding sites. So IgM has 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Whereas you go to IgGs and you have just 1, 2. But their number increases vastly as these numbers are declining between IgM and IgG. Okay, moving on. This is what I was talking about before. Here's your primary response. You get a certain amount of IgM, and as it starts coming down, you get finally the buildup of IgG. There's a time lag here. But secondary response, and this means secondary meaning second, third, fourth, fifth, 20th, you're going to see that you get a little bit of an IgM, but what's covering it on top of it and building up to a greater swelling amount is the IgG. Antibody levels in plasma, that's also referred to as titers. Okay, so antibodies use many different mechanisms to destroy target antigens. Neutralization is one. This is where the antibodies bind to the antigen, and they block the pathogen from entering the cell, such as blocking viral entry. Prevention of the pathogen adhesion. The antibody coating of the pathogen blocks adhesion to tissues, organs, things like that. Activation of complement. The bound antibody has complement binding sites, so this triggers complement binding onto the rest of the pathogen. Opsonization. When you start getting coated by antibodies, then complement proteins are present. They increase phagocytosis. They're kind of calling over the phagocytes like macrophages that are, come on over here. This is a target for you. Attraction of phagocytes. This is antibody binding, will attract eosinophils, neutrophils, and macrophages, which is kind of what we just mentioned. And then what also happens is stimulation of inflammation, because antibody binding promotes the release of chemicals from the basophils and mast cells, which will trigger inflammation, the inflammatory response. Now, a couple of other quick things about antibodies. We use them so many ways today. We'll mention about this in blood, also the blood chapter. Precipitation and agglutination. Now, remember, antibodies are soluble, but they can form these extreme complexes where one binding site of one of the antibodies binds to one antigen, and the other binding site will bind to a neighboring antigen or blood cell or whatever. And so you end up forming these gobs, these complexes. And this is particularly helpful or particularly evident when you see this with large numbers of antibodies binding to antigen-determinant sites. You get this big collection or cluster of cells. And the complexes eventually start forming an insoluble complex. This is where you get precipitation. Now you see agglutination, the clumping of the target cells, and the antibodies. This is where you have the antigens bearing. If you remember, we talked about, or you've probably seen, blood typing. Okay? Basically, here it is. You want to know if you're A, B, or type O blood? Simple. You take antibodies against type A, you put a drop of that antibody mix into a well of a dish, and then you put also subsequently some of the red blood cells. If they start clumping all together, guys, those blood cells have type A antigen present. You can do this also with an anti-B antibody. And if you have type B present, you get clumping. If not, it just becomes sort of this nice soluble mix of drops. Now we're going to briefly go through hypersensitivity here. This is abnormal reactions to antigens. Now hypersensitivities, otherwise known as allergies, are excessive immune responses. There are about 4 different branches. You'll get into this much more in detail in medical microbiology, but one of the hypersensitivities is allergies. And usually when we talk about this, you have a sudden increase in cellular activity or antibody titers, and this is going to occur by exposure to an antigen. Now, allergens are antigens that cause allergic reactions. You have an immediate hypersensitivity. This is called sensitivity type 1. It's rapid, severe response to an antigen. An example is allergic rhinitis. If any of you suffer from, let's say, hay fever, that's a localized inflammation of the nasal membranes in response to certain antigens like cut grass or tree pollen or ragweed pollen, etc. But if it goes from a localized perspective, you might get anaphylaxis, which is systemic. This is where you have a circulating antigen that affects mast cells throughout the body. And what mast cells do will release histamine, and they'll just start kicking out all sorts of cytokines. You're going to have severe cases that lead to a drop in blood pressure, and this is because you're going to have an excessive peripheral vasodilation. When this vasodilation occurs, the pressure will drop, people will pass out, etc. That's where you need to get epinephrine in, which will do the opposite. Instead of vasodilation, vasoconstriction, and increase the heart rate, so it increases blood pressure and the pulse, but it keeps them alive, as opposed to the next step, which is anaphylactic shock. And this is due to severe circulatory collapse, massive peripheral vasodilation due to the antigen exposure, and it can be life-threatening. So here you have the first exposure, the antigen fragment, let's say allergens, pollen, and you go through it, activation of the T helper cells, which eventually lead to B cell sensitization, which lead to plasma cells and the release of, note this, IgE antibodies. They're a little bit different. When you have the subsequent exposures, you're going to move with a lot more activity because you've got sensitization of mast cells, basophil cells. You're going to have them binding to the IgEs, which will cause a release of histamines, leukotrienes, and other chemicals that can cause pain and inflammation. If it's a localized allergic reaction, oh, you got urticaria, which is hives, or you've got itching, or you've got watering in the eyes. But if you have massive amounts of histamines released, you have massive amounts of reaction, the allergen is in the bloodstream, not only are you going to have itching and swelling, but difficulty breathing. This is due to the airway constriction, and this is where you get into anaphylaxis. Now, epinephrine, what would that do? It would reverse airway constriction, causing airway dilation. It would also cause arterial blood vessel vasoconstriction to elevate blood pressure. Okay? Now, I want you to take some time to review the flow chart, which is where the innate immunity and the adaptive immunity work together to defeat the pathogens. as you can see here. I'm not going to reiterate it all, but I would encourage you to take the time, and you will see this in your textbook. Notice that there is some distinctive differences here in the approach with bacteria and viruses, in part because bacteria may be present more often outside of the cell than inside, whereas viruses have to get inside the cell as part of their life cycle or reproductive cycle. Okay. Now we're going to go into a few other little aspects of adaptive immunity, and we talk about disorders. Autoimmune disorders. This is where you form autoantibodies, in which antibodies against specific tissues or targets. You have rheumatoid arthritis. There are other types. Thyroiditis or type 1 diabetes mellitus. Thyroiditis, you have actually the immune system attacking some of the thyroid tissue. The arthritis form will be where you attack the cartilage and the serosa joints that you see, leading to inflammation and eventually to damage to that tissue. Diabetes mellitus, well, we've mentioned it a little bit before, but this is where there's a lot of evidence that suggests that there's an autoimmune disorder where the immune system starts attacking the beta islet cells and eventually destroys them, and as a result, you have a reduction in the production of insulin. Now, transplant rejection. This is where you need immunosuppression drugs, such as cyclosporine. And what do I mean by that? If you see here, they're getting ready to transplant this organ in. Now, one of the things that happens with transplant rejection is that you take as close as possible an organ or tissue and you put it into another person's body. If you match most of the antigens that are keys, which are MHC factors, and you also, of course, bring in other factors like, what's the blood type, etc. You try to match them as close as you can, but in some cases, especially in the past when they didn't have such a good matching of these characteristics, the immune system would detect the foreign tissue and start to destroy it. One of the first heart transplants done by the Dr. Christian Barnard, he transplanted into a gentleman a heart, and it was pretty evident within 7 days that the heart was already being rejected, where the immune system was already sending in white blood cells in between the cardiac cells and destroying them. Okay. This drug, cyclosporine, suppresses or dampens down the immune system to reduce the chance of transplantation. The other thing, though, that you got to keep in mind is that people who are on immunosuppressants are much more susceptible to other types of infections, etc., and they have to be basically cared for or monitored. Flu can be serious, or other types of infections can be serious as compared to somebody who has normal immunity. We've already talked about hypersensitivity with allergies. Then we have the other issue, which is immunodeficiency diseases. Many times these are genetic. They're a malfunction of the development during embryological development. And that's usually one way that you're going to have immunological deficiencies or immune deficiency diseases. You can get it from some pathological agents. The best known is HIV, AIDS, and basically the virus suppresses some of the immune function. You can also have suppression of immune function because of exposure to immunosuppressive agents such as drugs or radiation. So some individuals who are on chemotherapy, some of them are on radiation therapy, they have to still be monitored because of the fact that they may be susceptible to other infections because their immune system has been blunted down. Okay. Finally, you have the age-related reduction in the immune function. As we get older, you will have a reduction in the thymus. We've talked about this. The thymus will, after puberty, begin to atrophy. You will have the cells being replaced by more connective tissue and fatty tissue. As a result, we'll also see a reduction in thymic hormones. and the helper T cells numbers are reduced. The B cells are less responsive, and you have lower antibody numbers. There is, later on in life, the enhanced risk of death by infections or a higher risk of cancer. And all of this comes back to the immune system. Higher risk of cancer because the immune system is not as sharp in detecting the development of cancerous cells and thereby can't catch the developing cancers quick enough to effectively eliminate them. Death by infections, same situation. With a weakened immune system, one is much more susceptible to infections that normal, healthy, younger persons would throw off, would bounce off very quickly or fight off and go on with their business. But older individuals, these infections could cause much more damage or actually threaten their lives. Okay, so let's move on. Now we are at the end of the chapter. I encourage you to peruse the chapter review outline on pages 791 to 793. Complete the review questions on pages 794 to 795. Complete the review sheets on pages 764, 774, and 790. The next chapter that we're going to approach is on blood, and I hope you all have a wonderful day.